and hello Richard. Welcome to the latest interview in the Down the Rabbit Hole series. Today we're going to be talking about trees, yes trees, and how genomics is being applied in this often underappreciated area of research. So Richard, if you could just start off by introducing yourself and telling everyone a little bit about what you do as well.
Hi, thanks Shannon. Yeah, my name's Richard Buggs. I'm a senior research leader at Royal Botanic Garden Kew, and I'm also Professor of Evolutionary Genomics at Queen Mary University of London. And as Shannon says, my research is mainly focused on the genomics of trees.
Yeah, so one of your kind of main areas of research is kind of looking at how genomics can be applied to understand the basis of pest and kind of pathogen susceptibility in plants. Why is this area of research so important?
Well, it's really a growth area because unfortunately, globalization is spreading pests and pathogens around the world very fast, not just of humans, but also of plants and animals. And we're really focused on the diseases that are coming and hitting our native tree species in the UK. And unfortunately, there's more and more of these pests and pathogens coming in. We're moving them around the world through global trade. And then climate change also is exacerbating the problem. So this is unfortunately a real growth area where more and more research is needed. I guess in the past, we've tended to think that trees just take care of themselves, but unfortunately, increasingly, we're seeing that isn't actually happening. You know, we lost elm trees back in the 1960s as large trees in the landscape, and more other tree diseases have been coming in over the last few years.
And kind of one of your biggest projects is looking at the genomics of ash tree species. Why are trees kind of in general so kind of difficult to research in terms of their genetics?
Well, classically, when looking at plant genetics, people would be working on plants with an annual life cycle, or even a life cycle that can be got through in just a few weeks, like the model plant Arabidopsis. And so in those situations, it's very easy to cross plants and then raise up a family from them and do a genetic map and maybe do another family of another generation. You can also make recombinant inbred lines. So you can have a line that's got very little heterozygosity in it, and you can just study particular loci that you're interested in. And of course, we can't do that with trees because they live so long. They don't start reproducing till they're several years old. They're massive. We can't just grow them in a growth chamber. So in many ways, working with trees is actually much more like working with humans than like working with wheat or barley or the model plant Arabidopsis. And so we have to start thinking, well, are there methods that have proved useful in human genetics that we can use on trees? You know, because like trees, you can't set up controlled crosses of humans that you decide you want to cross. You can't raise up a generation in a year. You know, all of the problems that we have with trees, we also have with humans. But of course, there's been massive amounts of funding for research on human genetics. And so we can benefit from that and pick up on many of the methods being used on humans and apply them to trees.
And how have some of these kind of advanced technologies kind of helped with some of these challenges and allowed us to kind of gain more insights in this area?
Well, it's made issues that in the past we just could not have tackled in a short space of 3 or 4 years into issues that we can tackle in that kind of time span. So, for example, my group wanted to find the genetic basis of resistance to ash dieback in our ash trees in the UK. We knew that some trees seemed to be surviving the fungus, and studies in Denmark suggested that some of this had a heritable basis, so that there is a genetic basis for resistance to ash dieback. And so we wanted to know what genes are actually involved, because if we know what genes are involved, then potentially we could accelerate breeding programs by having a genotyping platform to select trees with the alleles that we want to breed into populations. You know, if there are lots of different alleles that are all contributing a little bit to resistance, if we can bring all of them together in a single lineage, then that hopefully will be a more resistant tree. So in order to find those genetic loci, we of course couldn't set up crosses between different ash trees and then wait for the progeny to grow up and then test them for ash dieback resistance. That would take literally decades to do. But using approaches like genome-wide association study that have been developed very much with humans in mind, and also techniques of genomic prediction, which have been developed for livestock like pigs and cattle. Using these approaches, we can actually make much more rapid progress in discovering the genetic basis of resistance than we could have done with a linkage mapping approach. So they've really helped us in that way.
What is the kind of current landscape of ash dieback, and why did you kind of specifically focus on ash trees?
Well, ash dieback is getting worse and worse at the moment in the UK. All over the country now, we can see trees dying back and dying off completely. And the disease, this fungal disease, was first discovered in the UK in 2012, although it was probably here before that. And it spread across the whole of Europe, spreading from Poland westwards for the last 20 or so years. And it's really devastated ash populations throughout Europe and in the UK. So we actually started working on the ash genome in 2012. The Natural Environment Research Council gave me a grant then to do the first genome sequence of a British ash tree. And since then, we've built on that with bigger studies looking for resistance alleles, as I mentioned. And, really, if there's been any doubt in my mind whether or not this is a worthwhile problem to look at, this year has really confirmed it, because the ash dieback damage I've been seeing around the country this year is actually so much worse than it's been in the past. And woodland owners and the Highways Agency and Network Rail are having to spend huge amounts of money removing diseased ash trees so that they don't fall over on top of cars or on train tracks or on people. They're a major health and safety issue now, because the pandemic has got to such a stage where lots of trees are dying off.
This may sound like an obvious kind of question, but why is it so important to ensure that we do kind of control this?
Well, if we don't do something, we potentially face a situation where we no longer have ash trees as a tree for planting in woodland forestry. Indeed, we're sort of already at the stage where people don't really want to plant ash. And at the worst extreme, we could end up with no ash trees in the landscape, a bit like what has happened with elm. There are now very, very few elm trees in the British treescape because of Dutch elm disease. And some of our research has been on this issue. How bad could it be? Could we lose all of our ash trees? And the results are encouraging in that we don't think we will lose all of them. There seems to be enough resistance out there. And if our genomics work is correct, then it suggests that natural selection will be able to kick in, perhaps is already kicking in. And in the younger ash trees that are coming along, we are seeing a higher frequency, we hope, of alleles associated with ash dieback resistance. And so in some ways, the genomics work has given us a bit more optimism about ash than we had for elm trees, although things still look pretty dire. And this isn't really a case of being able to control ash dieback. That's completely uncontrollable at this stage. It's about thinking about the future and how we can ensure that we do have ash trees in the landscape in 50 or 100 years' time. So we're definitely going to see a big dip in the ash population, but we're hoping it will recover partly through natural selection and also perhaps partly through a breeding program. If we decide as a nation to do a breeding program for ash, then we could use the genetic markers to rapidly breed ash trees with greater resistance to ash dieback, and perhaps do that much more quickly than natural selection will be able to do it.
Are there any sort of kind of like driving forces that are kind of increasing the spread of ash dieback? And is there anything that humans are doing that kind of exacerbating that kind of spread?
Well, we definitely exacerbated its spread in the early years by the way we source saplings for planting. So a lot of our ash trees for planting were sourced from the European continent. So they were sold as British ash trees because they'd come from seed from Britain, but those seeds were exported to the continent, and then they were grown up on the continent and then brought back into the country. And many of them were coming back in with ash dieback already in them. So there were definitely things we did that increased the rate of spread of ash dieback. But on the other hand, it is a disease that is spread by spores that are wind-blown, and it was inevitable. Once the European continent was infected, it was really inevitable it would come here, it would blow across the channel. And so all we did was accelerate the inevitable, but it would have been very, very hard to have actually stopped the pandemic from reaching Britain, unfortunately.
Yeah, you work alongside Navagene, and I know that you mentioned that you've been doing kind of like genome-wide association studies and things like that. What are some of the other kind of specific genomic tools that you are using, and how are you being able to kind of track this resistance kind of that's kind of being like spread?
Yeah, so we've been doing a lot of whole genome sequencing of different individuals with different levels of resistance to ash dieback. And one method that we used to save money was a pooled approach. So rather than sequencing every individual separately, we put our ash trees into pools that came from the same seed source and had the same level of resistance to ash dieback. And so for every seed source, we'd have about 40 more resistant trees and about 40 more susceptible trees. And then we would sequence them all together to quite high coverage, but to much lower coverage than we would have had to have used if we had done them each individually. And of course, we didn't have to pay for the library prep for each individual either. We just had to do one library prep per pool. And that enabled us to estimate allele frequencies in the different pools. And we could do a genome-wide association study based on those allele frequencies in the pools. So that saved us a lot of money, but got us quickly to candidate genes, which is what we were wanting, of course. And then we were able to use those candidates to do genomic prediction on trees that we had sequenced individually. So that's the main approach that we used initially. More recently, we've been studying a natural woodland in Surrey in the UK, and we have been doing individual genome sequences for each of those trees and predicting how susceptible they are genomically to ash dieback. And so in that next stage of the work, we have been doing individual whole genome sequences. It really depends on how much money we can get at any particular time, of course, and on the drop in the cost of sequencing as well.
What have been some of the kind of key findings, and how will these findings ultimately help kind of overcome ash dieback and kind of help the kind of species that kind of live, I suppose?
Well, we're very much hoping that these results will be able to inform a breeding program. If we're able to grow up lots of ash trees and then at a seedling stage sample their DNA and genotype them and then predict which ones of them are worth growing on, then we could save time and space in a breeding program, which for trees, of course, does take up a lot of time and space. What I'd really like to do is develop lines of ash that flower very early, and then we can do something called rapid cycle breeding. This is something that's been done in fruit trees. So apple trees have been engineered so that they flower at a very young age. And so then you can take them using marker-assisted breeding, you can take them through lots of generations of a breeding program. And then in the final generation, you segregate out the transgenes that are causing early flowering. And you end up with a tree that's not genetically modified, but has been through an accelerated breeding program that has used its natural genetic variation. And if we could do that on ash, that would really accelerate breeding programs and enable us to develop resistance to ash dieback, increased resistance to ash dieback much faster using natural variation than we would be able to otherwise.
And what are some of the kind of key challenges that still kind of exist in your research?
Well, the major challenge is just the longevity and the size of the trees. One issue that's a real challenge to all genomic prediction methods is how specific they can be to particular genetic backgrounds. So just because we found a set of alleles that are predictive of ash dieback resistance within the UK using the sample we have of trees, transferring those markers to other situations isn't always straightforward. If we could find alleles that are always predictive of ash dieback resistance in any genetic background, then our results would roll out really well across the whole of Europe. But unfortunately, at this stage, we are struggling with this problem of the extent to which genomic predictions are genetic background context specific. So that's like with any use of genomic prediction in breeding of livestock and crops, this is a big issue that we face.
You also work on birch and oak trees as well. What are you exploring in this area, and how kind of different are they, like in terms of like the challenges with working with these different species?
Well, with oak trees, our main emphasis is on trying to understand a complex syndrome of oak trees called acute oak decline. And it's a syndrome where the trees start to bleed in their trunks. They have this sort of black goo comes out of cracks in their bark. And some trees recover, but some trees die of it quite rapidly. And it's particularly common in parklands around big country houses, which of course are quite common in Britain. And that's a major concern for oak, which is our most important broadleaf species. Because it's oak, we've been able to get enough money to do lots of whole genome sequencing of individuals. We haven't had to take a pooled approach. And at the moment, we're just gradually building up a really big set of whole genome sequences so that we can do a genome-wide association study. At this stage, we don't even know if there's a heritable basis to resistance to acute oak decline. But conventional methods of working out if there's heritability would be really difficult to implement. So we're actually trying to do a genome-wide association study to work out if there's heritability of resistance or not. You know, normally you would have answered that question first, but because it's oak trees, we can't do that. And so we're having to take this genomic approach just to work out whether or not there's heritability of resistance.
And what about your kind of work in birch trees as well?
In birch trees, we've had a big focus on dwarf birch, which is a nationally scarce species. It's a shrub, and it just grows on mountains in Scotland and one or two other isolated places. And it's been declining for many centuries, and we've done work to understand the genetic variability found in populations of dwarf birch, and where it might be good to plant different genotypes of dwarf birch in the future, and which populations would benefit from assisted gene flow. So that's much more of a conservation population genetics type study that we've been doing on dwarf birch.
How can all this kind of research in trees help with kind of climate change and sustainability and things like that?
Well, obviously, there's a massive emphasis at the moment on tree planting, not only in the UK, but around the world, to sequester carbon. And of course, we need a diversity of tree species to be able to plant in new woodlands. But if pests and pathogens are decreasing the palette of tree species that we have available for planting, that of course means that we end up planting woodlands that are less resilient, less diverse, less able to support a range of invertebrates and other biodiversity. So if we can tackle these tree health issues, and if we can keep planting ash and maybe even bring back elm so that we can be planting elm as well, we just end up with a more diverse, more resilient woodland in the long run. And of course, every time a new pandemic sweeps through, a whole load of carbon that's fixed in trees becomes released because the trees die and then they rot or get burned. And so this is really key to, trying to sequester carbon and trying to keep carbon sequestered that has already been sequestered.
Yeah. Kew actually launched a project called the UK National Tree Seed Project in 2013. What is the kind of ultimate goal of this project, and kind of how will that help with conservation and research as well?
Yeah, so that was a major project that was seed banking seeds from native UK tree species all across the country, sampling trees across their full range in every native seed zone across the country and at different altitudes as well, and banking them in the Millennium Seed Bank at Wakehurst. And so that has created a store of the genetic diversity of UK tree species. And it's something that we can hopefully go back to if we need to after a pandemic. But also it's an incredibly valuable resource for someone like me who's doing genomic research, because every so often we have to take seeds out to check they can germinate. And so what we're doing now is starting to do genomic research on those seedlings from these collections in order to try to understand signatures of local adaptation across the country in these different trees. And if we can do genome environment association studies on these seed collections, then we can start to make suggestions about what seed would be best to plant in different areas of the country. So at the moment, we have quite a crude system of deciding what to plant where. We have the country divided up into different seed zones. And if you're someone with a grant from the government to plant a new woodland, you have to plant seeds that come from your seed zone or from one of the neighboring ones. And that's how we're trying to ensure local adaptation. But of course, that's a very crude measure. And if using genomics, we could actually be saying, well, look, this seed source is very well adapted for this type of environment and this one for this type of environment, we could have a much finer and more detailed picture of what needs to be planted there. Or indeed, if there's very little local adaptation, then we could say, well, it doesn't really matter where you source your seeds from. You can get them from anywhere in the country and plant them, and then it becomes less restricted. And also we can start to factor in climate change and say, well, let's predict what the climate will be in 20 years' time, and let's plant seeds with that in mind and include that in our models. So we're hoping that this research will end up making our woodlands more productive and more able to sequester carbon rapidly.
Yeah. I mean, aside from kind of your research in trees, you also investigate food insecurity in Ethiopia. Would you be able to kind of expand on kind of what you're doing over there and the work that you're kind of looking into?
Yeah, so I'm collaborating with a much larger consortium that involves Royal Botanic Gardens Kew and University of Greenwich and many different institutions in Ethiopia. And we're all working together to try to understand genetic signatures of local adaptation in crops grown by farmers in the southern highlands of Ethiopia. And it's a really fascinating area. You have huge altitudinal gradients and highly productive farms. This part of Ethiopia didn't have the big famines that made the headlines back in the 1980s because they have very resilient food systems. And so we're trying to look for signatures of local adaptation in the crops and the different plants that are being grown by farmers to try to help understand what should be planted where and how that should change under climate change. And one of the major focuses we have, and this is led by a research fellow called James Burrell, one of his major focuses is a plant called Enset, which is a massive plant, a relative to the banana, that's grown not for its fruits, but for its stem. And the stem is processed and eaten. And we're doing research on local adaptation in that and how many different varieties there are and how planting can be optimized for that particular crop that supports over 20 million people.
And what are some of the kind of key things that you found so far?
Things that we're at quite an early stage with this, but in terms of the work on Enset, we found that there are lots of different lineages, but sometimes it's hard to tell from the names that are given which one is which. So there are some varieties that have different names but turn out to be genetically very similar. Then there are other things that have very similar names but turn out to be genetically different. So that's been very interesting. And of course, part of this is due to the the highly local nature of planting for some of these strains. And then going forward, we want to look at things like disease resistance in NSET, and as I say, that the degree to which it's adapted to the altitude, but we don't have any results on that yet. We have the data that we're analysing, but don't yet have results. So watch this space.
How are you collaborating with the kind of researchers and the local farmers in Ethiopia to kind of conduct this type of research?
Yeah, we have really strong collaborations with universities in Ethiopia and really good relationships with individual academics out there, and they have some really good collections of different races and strains of the different crops that are being grown. And so we're sort of helping with genomics to try and sort of genomify this research, and we go around to local farmers with them and interview the local farmers about what they're growing and ask them the names of the different strains of the different crops that they're growing, and try and understand whether they've bought the seed commercially or whether they produced it themselves. So it's really fascinating work, and as I say, highly collaborative with Ethiopian farmers and academics.
It's a big team effort.
It is a huge team effort, yeah.
I mean, this is the ultimate question, but kind of what does the future for you look like in terms of kind of research within the kind of tree area?
Well, unfortunately, it's a growth area because there are so many tree diseases, but also because we do now have the ability to do something about them through genomics. And I'm quite optimistic that in general, there is a lot of potential to improve our trees in terms of their productivity and their ability to fix carbon. Because if you just think about a crop like wheat, and you think about the wild relatives of wheat that our wheat came from, and then the extent to which we've been able to improve the productivity of wheat over the years through breeding. And then you compare that with trees, where essentially trees are undomesticated. We've not really done any breeding with them, especially our native broadleaf trees. And then I think, well, if we've been able to do what we've been able to do for wheat, what might we be able to do for trees if we really start to put effort into tree breeding and accelerate it with genomic methods? You know, this now I'm not talking about doing GM. I'm just talking about traditional breeding programs, but using genetic methods to know what's out there and to select the trees at a very early stage. And I'm very optimistic that we'll be able to improve the growth rates of trees and the growth form of trees and the speed at which they can fix carbon, and maybe even the size to which they can grow. And if we could do these things, then we could really have a much faster carbon fixation and woodland growth and timber production, and just be more sustainable in the way we use natural resources.
Yeah, thank you so much for joining me today, Richard. I mean, I feel like with genomics, we often we look at it from the aspect of kind of human health, but I think obviously our environment around us is so important and obviously affects us. And I think trees are such a big part of our environment, and this research is really important to kind of like understand that a bit more and kind of help with things like climate change and sustainability and things like that. So thank you so much for sharing your insights and the great work that you're doing as well. So thank you.
Pleasure. Thanks for having me.
Hello everyone, if you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website frontlinegenomics.com. I hope you enjoy.